Right. Yeah, it's really my pleasure to introduce my colleague, well, our colleague, and my close collaborator, Marinetics endowed professor at SoMAS, Bassem Allam. Bassem is responsible for me still working on the Thraustochitриds because he introduced me to those organisms when we both arrived in around 2003, coincident with the outbreak of QPX disease in the Raritan Bay hard clam population. And we worked together for years on QPX and eventually gave the thing a real name, which was terrific. So Bassem, I'll go ahead and tell you his academic history because that's what we're supposed to do, right? Has master's degrees from Lebanese University, master's and PhD from the University of Western Brittany in biological oceanography. That part I actually didn't know. Yeah. Then postdoc in molluscan pathobiology. He's done aquavet and lots of other things. He's really a world leader, not only in the disease ecology, physiology, and immunity of bivalves, but also in bivalve genomics and marker-assisted breeding of strains. And I assume we're going to get to hear a little about that today, which I don't know much about his work on that, so I'm really looking forward to it.
Thank you, Jackie. You hear me fine in the back? Perfect. Thank you for the nice introduction. Thank you all for being here. Indeed, I will be talking a bit about some aspects of our work that many actually in my here in the school may not be very familiar. Our background, I mean, our lab is called the Marine Animal Disease Lab. Indeed, this is the bread and butter of our activities. We do lots of things on pathology, immunology, host pathogen interaction. But frankly, today I'll not be talking a lot about those. I'll be more talking about these animals' ability to respond to challenges, whether they are pathogenic or environmental, and their ability to acclimate to these conditions, but also to adapt. So the potential of adaptation for these organisms, and this will bring us to genetic diversity and how they can respond to these challenges. So you see here my affiliation, of course, but I want really to acknowledge the support of the state DEC, who is a supporter of my own position here at SoMAS. So challenges and changing environment, many of you in the room are certainly more familiar than me with the left-hand side of this slide, right? You know that the environment is changing, many things happening in parallel, and so there is interaction all across these factors. So direct physical-chemical alteration related to climate, into temperature, dissolved oxygen, pH, salinity, sea level rise. We have in parallel habitat loss that can be related to these previous factors and so on, disease outbreaks, ontologic bloom, overfishing, eutrophication, etc., etc., etc. Lots of things are changing, and you know that. So how these animals respond and features that can be related to their resilience are listed on the right-hand sides. Physiological responses, plasticity, yeah, this can give them some acclimation. Genetic diversity can lead to adaptation long-term. So these two are really linked, but also population connectivity can gene flow, move around in a given area, yeah, that will also favor resilience. Of course, we can have restoration and conservation effort, and this is usually more productive when it's targeted, when we know basic biological information about the population we are targeting. That links back to genetic diversity and connectivity. And in many species, and actually currently all the species we are working on in the lab today are aquaculture. So being aquaculture, you can more easily manipulate things, you can grow things, you can culture things, you can select things. So the focus today is going to be on the boxed areas. On the left-hand side, we'll be giving an example of what we've done on temperature and pH, and of course disease. On the right-hand side, practically we'll be covering a little bit, tiny pieces of all those things listed on the slide. needless to say that these alterations in the environment, yeah, when we say environmental changes, have resulted in major disease outbreaks. There is a really good book here by a colleague at Cornell University that give examples and the author's perspective on these alterations. I recommend if you can read it. But clearly, the environment affects everything. Yeah, these animals cannot adjust salinity of their body. Shellfish again, they cannot affect alter or control temperature. They are homeotherm. So really, they are directly impacted by the surrounding environment. So in a theoretical framework, I presented here what would be host performance. Yeah, could be growth, could be feeding activity, could be anything. But the example here I gave immunity. Again, think about it as theory. Yeah, it never looked like this as simple as this. And on the other axis, the y-axis on the other side is the pathogen performance. Yeah. And these pathogens, remember, they can be what we call opportunistic pathogen, taking advantage of a weakened organism, versus obligate parasites, and can be pathogen as well, that are more specialized, that have long-term interaction with their home. But in general, these outbreaks arrive when the pathogen is superior to the host ability to control it. Yeah. And these are all altered by the environment. So you have really a complex interaction going on. I'm excluding here the microbiome and other things that we know they exist and control things. Yeah. This is a simplified view of those interactions. So everything is dynamic. Yeah. Which makes it challenging and interesting to study. Yeah. So a few examples from past history here in New York. If you're not familiar with the New York fisheries, historically, the biggest fishery used to be, for lobster, the eastern oyster. Yeah, I'm talking the 19th century. Oyster was really the ground zero of oyster production in the US was in New York. Yeah. Of course, in the 20th century, second half of the 20th century, really around the 80s and 90s, there was a boom in production in lobster. And this map shows you, it's a relatively old map, it stops in 2010 maybe, shows you population abundance of lobster. Clearly, this is a case study, really, of species, what you call migrating north, yeah, deeper and north to escape high temperature level. Again, the American lobster is a cold water species, so we had a huge fishery here in New York. The older people here still remember it, but some of you may not know. A few miles here north in Long Island Sound, one of the most important fisheries was lobster fishing in Long Island Sound until the late 1990s, where a series of events led to the collapse. These events include severe mortality, associated actually. In that mortality events in 1999, 100% of the lobster that died had an opportunistic infection in their brain. Yeah. So again, these pathogens are always, in many cases, are always in the environment. As soon as they have a host that is impacted, weakened somehow, they take advantage and flourish. Yeah. So other problems we saw, like here, the second picture down, is what we call shell disease. The lobster, these cigarette burns on the carapace. This can be as severe that you see the meat through it in a live lobster. Yeah. So again, this condition, we only saw it in southern New England in the early 2000s. And now we started seeing it in southern Maine, and prevalence is increasing. So those species, cold water, at the southern end of their distribution are under pressure. Yeah. And they are suffering indeed. And you see it in the population abundance. We see it clearly, and we have data on that, reduction in lobster immunity, emergence of opportunistic disease like this one, which is polymicrobial. We still don't have a single pathogen associated with these conditions. And we have emergence of even non-infectious diseases. Yeah. So alteration not related to microbes. Another example, which is also very local, surf clam, which was the biggest fishery in the state of New York in tonnage, the landing, mass, not in money, relatively cheap. All the soups you buy is usually surf clam, like a clam chowder and a Campbell soup is surf clam meat. And these, here we have the production estimate in 2002 and 2012 in New York. Yeah, this is really the coastal fishery in New York. While here are offshore fisheries from federal surveys. So reduction also, and actually when you look at the surf clam, we have some data also on event of recruitment. The recruitments here are listed by these arrows, highlight recruitment, and the plot actually is what you call temperature anomalies. So if you look at this plot, you have recruitment only when we have negative temperature anomaly, meaning cooler environment than the average throughout the whole period. Yeah. It's directly linked to prevailing environmental condition, and actually the physiology of these organisms is demonstrated also to be driven by environmental condition. Here we have a study actually published earlier this year by Ray Shaya, who was a PhD in the team, who demonstrated direct impact of temperature on larvae survival. Survival is higher with the blue color, lower in the red color. Yeah. And here we have temperature on the y-axis and food concentration. This is lab experiments on larvae. So of course, the more food you give, the better the animal usually does, usually. That's not very surprising. We can get back to this a little later. But yet again, survivorship was really highest at relatively low temperature, and really the larvae did really badly at temperature above 25, 24 degrees. The same for growth, where we have a sweet spot here and decreasing growth at higher temperature and lower food, of course. Yeah. A similar trend, different metrics in adults. Yeah, surf clam as well. This is a little older study done in the lab where animals, surf clam adults, were exposed to two temperature regimes, ecologically irrelevant. Yeah, we see them in the environment. And actually what we notice is that food uptake, so filtration, ingestion, yeah, and assimilation, decrease at high temperature, while energy consumption, like oxygen, ammonia excretion, increase. Yeah, you tell me, yeah, not significantly different indeed, but when you look at the plot for growth here, which is energy available for growth, based on calculation of what the animal takes from the environment through food, what they burn, is really markedly lower at an ecologically relevant high temperature. Yeah. So the temperature is the main driver. Food matters, yes. How food you think would matter? We'll get back to this. Think about it. I'm talking to the students here. Why the food would matter. But oyster, yeah, I mentioned oyster. In the 19th century, early 20th, you could find, oyster on the side of the road, like hot dogs now in New York City, used to be oyster. Some other places in the world still do it, but not here in New York for safety reasons, of course. But look at the oyster production. Of course, I'm not present in the 19th century, but even from the middle of the 20th, severe decrease in production in New York, and associated with the appearance of diseases. Some of these diseases we don't know precisely when they started. Some we have a better idea when they started, at least in terms of outbreaks. So here are the date, and indeed this is a parasite that was originally discovered in the Gulf and was limited to lower Chesapeake Bay up until the 90s, and in the 90s really made a jump north, 600-700 kilometers north. Yeah. Clearly related to abnormal temperature regimes, an increase in particularly winter temperature. Winter temperature, the lowest of the year, were the limiting factor for disease spread. once these low values became acceptable for parasite persistence, it shifted north. Yeah. This one, the first one, MFX, again, a big problem for the industry. They just got a severe mortality outbreak in Canada. So they are migrating north. Yeah. All of them are migrating north. GOD was discovered here in New York, the really ground zero for this parasite. It's a bacteria, I could say. Juvenile oyster disease is called, and now it's a problem up throughout the Northeast, yeah, up to me. So there's really problems going on. I'm talking here about some work done by a few students. The students are all gone now, graduated. Michelle, Ray, and Caroline, some of you may remember them. On the impact of ocean acidification on biological health, of course, I mean, the obvious is that, yes, ocean acidification reduce the growth of calcifying organisms. Yeah, it's a no-brainer. Higher mortality, yes, of course, we also have higher mortality. Shell dissolution, you have here some mild cases of shell dissolution on the surface, but some severe cases. And these animals were still alive when you collected them. And of course, they limit repair of shell damage. So OA has multiple impacts. Let's dive a little further on impacts that are not as well studied, like this. Impact of ocean acidification on resistance to infectious disease. Yeah, this is really under-investigated. And Caroline in particular did a couple of studies. One in hard clam, one in oyster, where she showed significant increase in susceptibility to these bacteria, to Vibrio, when animals are also exposed to acidification. Like this bar here is significantly higher than everything else. And this is the treatment where the animal were exposed to acidification and bacterial challenge. Yeah, the same was found in oyster, where she also did, in addition to the bacterial challenge, transcriptomic and proteomic assessment, even cellular assessment of immune parameter, and she saw a reduction on all these immune factors. And Vibrio taking advantage of a weakened host and developing disease. So from here, why you think these animals exposed to OA had higher disease levels? Students, I mentioned a few slides back that will get back to some notions. Why animals exposed to OA will have, it's off? Oops. I'm working on it. Yeah. Any idea? Yeah.
So animals under stressors are going to have a worse immune response and then not have the ability to fight off.
Exactly that. Exactly that. It turns back to energy availability, actually. When you look at the energy they spend to fight this infection, this exposure to ocean acidification, they have a little less energy balance left over for immune processes. And this is, there are an easy way to demonstrate it, and the student did, was by looking at food and energy consumption. So bivalves burn more energy under OA conditions. Yeah, this is here, you have respiration, that these animals respire way more larvae, in this case, for oyster, way more under OA conditions, but also they burn reserves. Glycogen is like the starch in animals. Yeah, this is carbohydrate reserve, they burn. This is in adults. They burn lipids, protein, a slight decrease, but again, protein is the last thing you burn, right? Yeah. When you start burning protein, that's really bad already. They burn more energy, and actually, when you feed them more, they do better. So this is an example of experiment Caroline again, where she had two feeding regimes, one called high food and one she called low food. I believe this one like 10, 15% of hatchery standard procedures. Yeah. On high food, acidified animals had, this is mortality, had as much mortality as the low food control without acidification. And when you add acidification, the mortality is significantly higher. Yeah. And in terms of growth, you have the opposite trends. So both growth and survivorship are related to amount of food available for these animals under OA stress. So food matters. And actually, in fact, not just the quantity of food that matter, the quality of food here. It's really a measure that we use regularly in our lab about sorting efficiency. These animals, these bivalves, don't ingest everything they encounter in the environment because that's not very energy efficient. They try to pick the particles of high energy constants. Yeah, we've been studying this for years, you know, now precisely how they do it, but they pick up and choose what they want. Yeah, we observed that this sorting ability increase under OA, meaning they can adjust their fooding preference to pick up the most nutritional food as well. Yeah. So what happened to that energy? How is that energy used? Really clearly in animal under OA, you see upregulation of genes, overexpression of protein related to biomineralization and ion balance. So they use a good amount of that energy to produce the protein that they are needed for maintaining ionic balance and building shell or maintaining shell. Yeah. And in parallel, down-regulation of immune gene, hence an increase in infection that we saw earlier. Yeah. So Caroline really had fun punching holes in shell, looking at this, what we call extracellular fluid. It's a thin layer of fluid that contains blood cells between the shell and the matter. It's very thin, yeah, in these animals. And following ionic balance and pH in this environment, but also hemocycle, and homicidal in RNA sequencing and so on and so forth. As well as in the blood. This whole, you can collect blood directly from here, silver muscle, or here we collect this thin layer of extracellular fluid. And this is the profile, the transcriptomic profile. This is RNA sequencing, gene expression. Things that are overexpressed are shown in warmer color, underexpressed are in cooler color, bluish. And clearly, immune genes would be here, yeah, in the blue areas, while of the acid side, yeah, while biomineralization gene will be in the reddish areas. So they can adjust gene transcription. This is plasticity, yeah. This is phenotypic plasticity. I mean, these genes have function to produce the protein that are needed for specific physiological functions. And we looked at how some of this information, these proteins are translated at the organismal level. So Caroline measured extracellular pH in the fluids, yeah, in the blood, and in that thin layer of extracellular fluid where biomineralization takes place. So measure pH, she measured them 3 times in a day. And what's really interesting in this, I really wanted to share it with you. We have the control treatment. This is control and seawater. Yeah, the triangles are seawater. Control treatment, seawater. Acidified treatment, seawater. Yeah. So the treatment basically 7.3, 7.8. But look at the pH of the blood and the extracellular fluid. While the water is down here, the animals work hard to bring the pH high, yeah, in that fluid, the calcification fluid and the blood. So the pH of these liquids is higher than the surrounding environment. So far, so good. And actually in the control, they bring the pH down. What this means? That means biomineralization itself to happen, it needs a tight pH level in these fluids. So if it's too high, they bring it down. If it's too low, they bring it down. And this uses energy. Yeah. This uses energy. And actually, when you look, what we also did, into intracellular pH, the pH inside the blood cells themselves, not the liquid that surrounds them. Yeah. The pH actually is higher in the acidified animals versus the controlled animals. So animals under OA condition, their blood cells work very hard to increase their intracellular pH above normal so they can adjust the outside pH. Yeah. Good. All this is energy intensive. And actually, when you look at this gene regulation, you see clearly that gene regulation is consistent. Here, we follow the same animals in hard clam that were exposed continuously to OA condition or to control condition, to larvae, 2 days larvae. This is post-set, what we call post-set, meaning as soon as they settle, 32-day-old, yeah, and then 5-month-old animals and 10-month. And remember, these were exposed continuously to OA or not. Yeah. Gene regulation on a PC principal component value. This is really a indication of gene expression levels. Yeah, it doesn't tell you what gene was regulated, what gene was down-regulated, but how that genetic expression fingerprint shows. 2 days, we have separation between acidified and control, clearly. And this is consistent. Yes. Of course, gene expression at 2 days of larvae is different than gene expression of post-set. They are not the same gene that are operating for basic biological reasons. Yeah, it's different age. They have different functions. Yeah. But systematically, OA has an impact. And when you look what is shared among these genes, one of these genes is really pyrrolysine that systematically was overexpressed. Yeah, shown here. And actually, if we do silencing of the gene, specific targeting of the gene, shut it down or knock it down, actually, it's not completely eliminated, not CRISPR. This is not CRISPR based. This is RNAi, RNA interference to silence a specific gene. Actually, when you silence the gene, biomineralization decrease markedly in the acidified animal. But that was in the control. Yeah. Even animals under control water condition, not acidified, when you shut down the gene biomineralization is reduced. It's really a main player in that cell deposition process. But under acidification, really, it's a market impact. So we can zoom down into specific gene, what their function, which is really. So many challenges, yes, it's really the introduction so far about the focus of my talk. Many challenges indeed. And the main question is, can these animals adapt? Yeah. So adaptation again, we saw example of phenotypic plasticity, gene regulation, change in food uptake, and things like this. Yeah. The focus of the following half an hour is going to be on genetic diversity, because this can enable long-term adaptation. When we say genetic diversity, this is an example how that matters. Yeah. Here we have 3 clam strains. Strain 1, I'm not giving the source. Strain 2, strain 3, that we got at the same age, we deploy in the same environment, and followed a disease called Tripex disease. Jackie mentioned it a little earlier. And followed disease development. Strain 1 developed disease within a few weeks. The disease worsened 3 months later. A year later, we have this high level, 50% at least infected, and mortality was high in this. Genetic line of clam number 2 developed a tiny amount of disease during only the following year, not the first year at all. Strain 2 never developed the disease. This is, I think, a strong indication that genetic source of the animal matter a lot in disease resistance. The right-hand side, this has been published a while ago. The right-hand side is oyster. Here we have juvenile oyster. and how they fare in terms of mortality against juvenile oyster disease, bacterial infection, under OA acidification, OA conditions. So we put both stresses together. Yeah. OA and bacterial. And we have the control, of course. I'm not showing the control here for simplicity, but mortality is shown here in days. We exceeded 50% in this strain of oyster. Yeah. While this strain of oyster showed no mortality at all. Yet again, strong demonstration that the genetic background matter in terms of OA and infection. So genetics really matter a lot. So theoretically, we can have superior animals, yeah, in the population out there that can enable adaptation, long-term adaptation. So the animals have that potential. Do we have the ability to change our practices? And this is the we in my title. Can we adapt our strategies to build on this genetic diversity to improve things that it's where we can? So in other words, can we develop strategies to build on this genetic diversity to help conservation or restoration of natural population, but also for aquaculture stock to develop, identify and develop resistant stock for aquaculture. So when we say genetic diversity, there is a prerequisite for that, yeah? We need to know what we are talking about. How to identify genetic diversity if we don't have genomes that build on. And actually, many of the species are non-model species. So we have really to start from scratch and develop genomic resources ourselves. For example, this species, Barsenaria, we sequenced the genome here at the Allam with collaborators, of course. The paper was published a couple of years ago. Base scallop, the paper was published like 3 weeks ago. So we sequenced the genome. And now we have the background, the baseline information to build on to understand genetic diversity. Yes. Yeah, the bottom line here, I know Jackie would be interested in this. And I mentioned to her, she actually have the genome already. We also sequenced genome of parasites. This parasite affects this plant, and that parasite affects the scallop. So we have the host and the parasite, so we can zoom in really on interaction between the two. But I'm not talking about that. So when we say genetic diversity, there are many features of genetic diversity. This is the most common one, what we call single nucleotide polymorphism. Single nucleotide polymorphism are really one single nucleotide that change between an animal and the other animal in a particular location. So this is C, yeah? Because in this animal we have it as A, here a G, here a T. Yeah? This is a single nucleotide polymorphism. Bivalves are hotspot for this variation. But here I'm contrasting. Bivalves have oyster and clams. About one SNP every 20 to 30 days. A human have one SNP every 200 to 500. So they have big reservoir of this variation. What this means? Why these variations are important? You may remember introductory biology. These variations, if they are in a coding region, encode the messenger RNA, which in turn turns into protein. So one SNP difference, yeah, can lead to different amino acids, or even can change completely the structure of the messenger RNA. Could be tell the transcription to stop in given places, the translations, yeah, can encode a stop codon. Instead of the protein being that big, it'll be that big. Yeah. And this will have phenotypic impacts, yeah, on the physiology of the organism. An example is here. This is cattle, yeah, beef, normal, and Belgian blue. You know what is the difference between these two? It's a mutation in a single gene. The mutation here is not a SNP, it's a deletion of 11 bases. 11 bases are cut from this one, are not present in this one. In the myostatin gene, so we have a frame shift of the protein, yeah. And this is lead to a phenotype called double muscling. We are not trying to do the same with oysters and clams, trust me. But again, this is just to show you that tiny differences, 11 base over the genome of this guy is 2.7 billion base. 2.7, human or 2.9, 2.7, and 11 out of these 2.7 billion make this difference. Yeah, all this to say that tiny change can have really great impacts. I showed you this example earlier, remember, with oyster, GOD, and OA. Actually, we did genotype them to profile the SNP characteristics. So we generated a genetic fingerprint based on SNP frequencies. And this strain, I can tell you, it's blue here, that was really strong. This is a New York strain. It's right here. This is the one, yeah. iSNP, it's right here. Yeah. So genetically different from the other strain. I'm not saying why it's superior here. Yeah. I'm just saying that genetically it's different. We'll get a little more detail a little later, not on this same example, on a different example. So those SNPs matter. Here, different strains have different genetic fingerprints. But the question that we are really interested in is, are survivors within a given strain genetically different from those that succumb to the stress? Yeah. So basically, contrasting resistant animal within a population versus susceptible animal within the same population. This is schematically shown here, saying we have an average population, whatever that is. We may have, hopefully, resistant animals in it and susceptible animals in it. This is putative so far. If you expose it to the stress, some animal will die. Hopefully, more percentage of susceptible animals will die, yeah? So if you contrast, because in many studies we cannot collect these animals, to contrast genotype here versus genotype here, we contrast time zero versus survivors at the end, yeah? Again, I climb in the field, we cannot know when it died, collect the tissue. Possible, although there are techniques you can get for that. So, and we've been contrasting these like time zero animal versus survivors. I'll give you an example here. This published also a couple of years back on animals that survived QPX outbreak. This is QPX disease impact on clams. It can be really a mess. 80, 90, 99% of the animals can die in a population. So everything on the surface is dead. A lot of animals are in the shed instability. Contrasting genetic fingerprints between animals at time zero and survivors at the end. And you see that survivors are really separated from the remaining one, meaning genetically different. Again, I cannot tell you because of gene A and gene B here, although we have the information. Yeah? But genetically different. We have the same trend really in ocean acidification. This is in oyster versus clam, where survivors can have genetically different fingerprints than animals at time zero. Here, actually, it's not time zero survivors at the end. It's really acidified animals and controlled animals in parallel. Yeah. So we collect them at the same time, either exposed to distress or not. So there is genetic diversity. Animals that are resistant are genetically different. This gives us a tool, or at least a strategy, to improve, identify stocks and improve stock. Yeah. And this is something we did with hard clams. That since 2019, we are leading a collaborative project with the institution on the project listed here to develop genome-based approaches to improve stocks in hard clam. And while we have a big vision initially, we limited ourselves initially to two traits: QPX disease, which is still a big problem here in the Northeast, particularly in Massachusetts, I should say, and heat stress, which is a big problem for our friends in Florida. That clam died because of heat stress, yeah. And both, we believe, are genetically, at least QPX is clearly genetically dictated, and there is evidence that the temperature also genetically dictated. So we can develop tools, and the tool we decided to use here is what we call a DNA ship, a SNP array. They're genotype animals at high throughput, at low cost. And this technology, I mean, it's been used, of course, for correctional crop, for marine species, salmon, other fish, that allow you really to breed for any phenotype, growth, freezing survivorship, whatever, name it really, as long as that phenotype is linked to genotype. Yeah. And many phenotypes are linked to genotypes. So it's really boundless. So how we do that? First of all, we have the genome, yes, this is the baseline. But we need to establish the species diversity because we need to know what is the starting stock of SNPs, yeah? How many SNPs are there in the species? Of course, we cannot probe each and every SNP, but at least get a picture. So we collect animals from Maine to Florida, sequence them, whole genome resequencing, and come up with a catalog of SNPs in the species. Here we identified 300 million in hard clay, yeah, 300 million SNPs, single nucleotide polymorphism, that we had to filter down to be able to work with. And the endpoint was 66,000 that are distributed across the whole genome, which is important for genomic reading. And for you to identify them, some of them are in coded regions, some of them are intergenic. We included even QTX marker to use the SNP shape, the DNA shape, as also detection tool, and a diagnostic tool, at least attempting to do that. And that SNP shape really has been published again like 3 weeks ago, 4 weeks ago, maybe a month or so. It's really powerful because for each clan, we can determine 66,000 markers at once at relatively low cost, yeah, like $50 without labor. And it's really powerful to segregate the plant population. Here we have both wild plant in the middle here. We had only access to wild plant from New York, New Jersey, and from Florida. And we have aquaculture plants here and here. Colleagues in Florida have three population of aquaculture plant. On our end, we had also four actually. Groups of aquaculture plant that are reflected as USDA here. We'll get back to them. They are really interesting one. The USDA clans are clans that we bred here in New York based on a handful of markers through USDA funded project before. This current project is funded by NOAA. Really bold on those animals. We did what we call marker assisted selection. So picking up really a handful, like 6, 7 SNPs, and created the group from a meta population, genotype the root stock, and decide that these animals are expected to be resistant, these animals are expected to be susceptible based on previous information in genotype to phenotype, and read them separately and then test them. So we produced 5 groups under the USDA project. We pursued the work. We pursued the work under on 3 of them genetically, because they were the most interesting 3. But let's start with the wild type. The tool allow clear and easy separation between plant from Florida. Yeah, come on, it's very far, Florida. It's very separation by distance. Yeah, it's not unexpected. While in New Jersey and New York, they are really similar. And again, New Jersey here, one of the population is from the other side of Raritan Bay. And our clans were from the other side, on the New York side of Raritan Bay. Come on, the same population. We knew that. I didn't know the source of the New Jersey clan until later. My colleague told me. Boring. While the second population in New Jersey was further south, but they still clustered together. The USDA clans, the more interesting one in my view, because we have a phenotype. So we genotype D, E, and F. 3 subgroups that we bred initially based on genetic markers. So we produced a group D that showed disease levels of nearly 80% disease, susceptible. Group E, about 40% disease, and group F, about 10% disease. So we have a difference in the phenotype, yeah? And actually, when you look at their genotype, it's clear. D and E are closer together, still a little separate, while F is really very different. And again, the phenotype is really very different. Yeah. So we have linkage with genotype phenotype. Here it's really the survivorship that is negatively correlated with the prevalence, which we know. So there is something into it, and we then dissected, looked a little more carefully to DEF before and after mortality. So not just the baseline genetic difference. Again, group G had high mortality, remember 80%. And animals that survived after 22 months in the field were separate genetically, have different genetic fingerprint than animal at time zero. Group E had average mortality, we still have a separation. Group F, which had 10% mortality only, survivors are still genetically similar, which is not surprising, right? There was no selection. They didn't die. And now we are using this SNP array to predict. So not just link disease levels on previous sample to genotype root stock, and then decide that these animals, we expect them to be resistant or not, produce them separately and validate them in the field. The first round of genomic selection was done this year by our friends at Rutgers University. We plan another breeding ground in Florida this year, and here in New York, we'll do it next year. And validate ability to predict disease, yeah, or survivorship overall. Because in Florida, it's not disease. And we've been doing something similar, again, this is quick, in oyster, a few projects that were ongoing, including this last project here. Because here too, we produced, we are part of the Eastern Oyster Breeding Consortium. We produce a SNP array for breeding purposes as well. And we have currently in the field here in New York, genomically selected oysters, yeah, versus traditionally selected oysters side by side to follow survivorship and performance growth and so on. So Sam Gowen, the lead on this is our colleague at Rutgers University. While Neil Grant and Iraq, we are lead. So I'll finish really by an example that we've been working on over the last 3-4 years that exemplify the role of the environment and the genetic, and how the two together can lead maybe to a perfect storm, condition that can lead to problems. Let's, you are probably now familiar, at least some of you, that bay scallop been suffering here in New York lately, since 2019 pretty much. This is landing of bay scallop. Bay scallop was important before. Then in the 80s, HAB event in the Peconics hit hard the population, production went down. And really production picked up after intensive restoration effort by Cornell Cooperative Extension. Let's start here with mid-2000. The population landing increased until 2019 and then boom again. While the current boom, the current problem is not linked to HABs. So it's, this is where we were dragged to this issue and discovered, I mean, probably rediscovered parasite, because I think it was there already, that is affected these animals severely. This is kidney of scallop, and everything around you see here is a parasite. It's full of parasite cells. You see them here yet again, there. That hit the kidney and other organ. Again, I'm showing an example, leading to heavy mortalities. Those who want to learn more about the disease, it's here. And actually, part of this paper, Emmanuel, who's sitting in the back there, did a study of prevailing environmental factor with the focus on temperature. And she reported this trend, that the mortality rate in 2019 and since was associated with abnormal positive temperature anomalies, temperature higher than normal. So we ran experiments in the lab to follow seasonal development of the disease and mortality under standard condition. I mean, this is natural seawater as it comes from the field. And implement an O-line flow through either a chiller to cool it down a bit or a heater to warm it down a bit and see how disease develop and mortality develop over time. We're going to experiment 1, January to July. This is only heating because we cannot cool the water in January. It was already cold. And the chiller do not operate at very low temperature. Well, the experiment 2, April to July, heating and cooling. This is a picture of the experiment 2 where we have the cooling here. So it's really thoughtful. This is done in South Africa. Experiment 1 was done in South Africa. And this is the temperature we measure. Rather good. Yeah, you can control delta of temperature. Experiment 1, we attained 1.5 degree. Experiment 2, either, yeah, this should have been cool, blue. This should have been like gray, that's fine. This is blue, yeah, this is cooler. The blue curve is about 2.2 degrees cooler than the normal water in the middle. So we attained the deltas. We are kind of targeted, yeah. Looking at disease and survivorship, experiment 1, although the delta was only 1.5, we had mortality in both, I should say, but there is still a significant difference for a statistician here. 0.49 is significant, yeah. So really, animals held at the higher temperature died faster and more. And disease level, we have 2 techniques for the diagnostic. One is microscopy based, one is qPCR based, and both are really increased a little early. So the warming temperature lead to earlier disease development. We had the same with the qPCR. Experiment 2 yielded similar trend, although here the difference in mortality was significantly high. Yeah. So temperature, a tiny difference in temperature can lead to either faster development or slower development and mortality. And there is genetics. These are different groups of scallop, genetically different levels of disease and mortality, and we have the same. And similarly, we were able to demonstrate that a genetic difference between the groups. So animals that survive better are genetically different from animals that die here. This is a discriminant analysis based on SNP frequency. Yet again, SNP frequency. So animals that survive later are genetically different from animals that die here. So the genetic matter, and from there we are pursuing our adventure. On scallop, we plan on continuing to work to really establish genetic diversity of species in the area. Use genomic tools to improve breeding, which is feasible, we believe, and other stressors. Now the focus is disease and mortality, but you can think about it other stressors as well, like HEVs or. Conclusion. So many problems facing these species, and the environment is changing. So it's a dynamic response. We need to be adaptive, but they have high genetic diversity that can enable adaptation. And the idea is to use some of this genetic information and strategies to help these species, particularly cultural species. With that, I will finish by thanking many people, team members, collaborators, but also really Amelio crew here. Dennis, who's online, Sarah, who was previous postdoc, and Caroline. I would name these two, these persons only because the list is too long. Thank you.
I think we have 2 students signed up to ask the first question.
So I'm just curious to know, if you have looked into how changes in water quality or an increase in untreated wastewater have complicated the ability of shellfish to adapt.
We haven't looked into that specifically. However, yeah, I mean, many things can alter shellfish health, yeah, shellfish physiology. So depending on the wastewater you're talking about, if it's bacteria only, like a human bacteria, these animals don't care. They feed them. Actually, they can digest many of them, yeah. So depending really on the contaminant you're thinking. The clam population we have in New York, the best clam population on the east coast of the US, is in lower New York Harbor, where all New York City outflow ends, yeah? So Yeah, it depends on what's in the water. And the kind of things you are interested in. There might be some lethal effects, yeah, like things we don't really see, obviously, like on reproduction. No, there is lots of things we human use and end up in water that can affect reproduction, even sex ratios. So it's,
So lots of oyster festivals now. You eat them raw?
yeah, we are in our month, October, September, October. It's relatively safe, depends on how they were handled, yeah. But from the field, they are on the safe side for now. In January, February, we start getting noroviruses, so be careful on those as well. But from bacteria perspective, we are safe. If they are handled well. After fishing, yeah.
You mentioned you want to produce the clams more resistant to the stress. I'm curious whether they will, this will and their temperature preference, whether they will prefer warm temperature and migrate to lower altitude.
Just to make sure I understood the question. For the clam, we are currently breeding for two traits. Disease here in the Northeast and temperature stress in Florida. Yeah. So your question is on temperature, if I understood well.
My question is whether the general modified clams will be will prefer warm temperature and migrate to warm temperature water.
I'm not sure. I'm sorry. Do you mind if I get closer? I can hear you. Yeah, I'm an instructor here.
Yeah, and you want to produce the plants adapt to
High temperature.
high temperature, and whether this will change their temperature preference. And it to the plant migrate to
Oh,
higher.
excellent question. So whether we will be facilitating the migration of those clam genetic lines further north with the global warming, yeah, with the warming. We cannot rule it out. I should say, however, Florida, biggest clam industry is on the Gulf Coast, which hard clam is not naturally present. Actually, it was introduced from the Northeast in many cases, and mid-Atlantic to the Gulf Coast of Florida for aquaculture. Yeah. So in the first place, we gave them the clam. So if we get some back, well, maybe not a big deal. But it's a good question. You know what? Let the best win. If indeed these, as long as we maintain an eye on genetic diversity, you don't want to have animals that are highly resistant to temperature to succumb to our parasite here in the Northeast. And actually they do. Actually, if you get the clam from Florida, put it in our water here, disease develop really quickly. That parasite, Chipex disease, develop like crazy. Very, very fast, because they are not selected there. Yeah. They are not selected for that because the parasite is not present there. So there will be a balance, yes. Yeah.
So you showed this, what was it, the particle efficiency
Yeah.
selection
Yes,
efficiency.
yes,
And you showed that under OA conditions,
Yes.
that was enhanced,
yes.
which I find
Counterintuitive.
counterintuitive.
Yes,
So can you,
yes,
because it's more energy that is required. Why
Yes.
is that?
absolutely. But actually in these animals, they gain more energy from the food they ingest if they are more selective. Let me explain. The digestion in these animals and in the digestive gland, which are closed sacs, yeah. So they try not to ingest everything they encounter, because there's lots of mineral particles, lots of debris, not very high nutritional value.
Plastic.
They don't want to fill these organs with things that have low value. So they pick up and choose the one that have high value. And this and ultimately lead to better gain. I mean, we have even some mathematical estimation and
But again, I think that, so what is the first group, are they being lazy? Why wouldn't they do it anyways? Because that's really
indeed they
more energy efficient. Why wouldn't they do it anyway?
they do it in the first place, but it's increased. So yeah, here it's comparative. keep in mind, yeah, we are comparing animals at rest, they select. Yeah, but that selection, okay, which has some energetic cost as well. So it's really a balance between how much you want to spend to get the best food versus to do what you get because you're fun, yeah? And actually, that's sorting, it's a good question there. That sorting, we know it increases when the animal is developing gonad. And actually, we have papers on that. If they are developing gonad, they require more energy, high quality energy, they increase the sorting. Yeah, so you can adjust that, it's a part of the plasticity, but
There isn't much of a cost because they have to transport the particles either way.
yeah, I mean, the only cost would be the
It's like-
protein.
Just reversing and moving in a different direction.
Yeah, which is based on some protein specifically produced for that purpose, right? So they produce those specific protein lectins, right? That attach to the surface of the particles of interest to them. To move them in the right direction instead of projection.
But the energy is mostly ciliary motion. That's what most of the energy consumption is probably.
Yeah, well, I mean, listen, Jack. Protein production in a larvae is about 40%, I believe, of the energy that for the food. So protein regulation, so yeah, relatively, you would think about it, yeah, well, it's a small fraction of the total, but there is a cost to it.
All right, we have not really much time, so maybe if you can answer this question
Yeah.
that comes in from the chat just really quickly.
Oh.
Chris, so do you think there is an ability to select for tolerance rather than resistance that could also provide improved survival?
Yeah, very likely, Chris. As you know, tolerance, Chris did some work on the tolerance in oyster with us, his previous species student with us. Yeah, tolerance indeed, in terms of parasite at least, as you know, will not select for better parasite. Think about parasitism, for example. If you have an animal that really resists a parasite, meaning it doesn't allow it to affect, the parasite will develop strategies to still attack. So we are doing an arms race between the two. If it's resilience, we can have still the parasite infected, complete the life cycle and so on, but the animal is surviving, not suffering too much. This is the perspective that Chris is coming. Yeah, it's a good question. We don't know, really, frankly, Chris, about those energetic balances and the cost and for these and many examples other than your work in oysters.
Okay, thank you, Bassem.
Yeah.